vitamins and detailed structures. Amino acids become content of them when they are broken down, and then the monosaccharides could be there, and the fat is going to be divided to smaller molecules. What then happens that all these are being resorbed in the small intestine, and then water is being absorbed in the large intestine. So this is the basic division, you would say. Definitely your digestion process starts in your oral cavity with the help of saliva, right? So that background knowledge आपको clear होना चाहिए with the help of previous things as well. So here we are supposed to study the histological feature, but definitely we will discuss few gross features to understand the things in clear. So let me select another picture. Okay, that stomach will be showing you various parts. The area just close to the esophagus is the cardiac region. Then you observe the dilated upper part is the fundus, and the whole portion is the body. Then you enter the pylorus, that has got the entra and pyloric canal. Then we see this C-shaped structure, the duodenum comes, the part of the small intestine. So it means the esophagus and duodenum are there, in between you get the stomach. These are the portions. And definitely we mentioned that there are 3 types of glands which are being named according to the location. Like cardiac gland, the fundic gland, जो body को भी cover कर रहा है, then the pyloric gland, right? So the regions are clear in your mind. What else? Now we are looking into the cut section of the stomach. Yes. So with the first look, when we see our few, you can see the structure that is very much clear are so many rather. Rugi, what are they? They are temporary foldings of the mucosa and the underlying submucosa, right? You can observe with the hand lens, or you can see with the naked eye as well, that it is just like a crumpled towel located inside. Is it clear? It's not a plain looking surface, rather in the folding you can observe. A look of that of sulci and gyri of the brain, isn't it? So now we are supposed to see next to it the esophageal sphincter here with the help of a muscular layer, and the pyloric sphincter, because we know that there are the muscular cisterna. So the middle layer is going to be producing this sphincter, right? Because it is well developed here. Okay, let's see some other related picture to see what are the 4 layers of your GIT.
Mucosa.
Yeah, mucosa, the innermost, submucosa, muscular cisterna, adventitia or serosa. Yeah, okay. So let's start first with the different glands. Okay, we observe the area where we saw the cardiac region of the stomach. Keep in mind. Now, if we cut that particular area, that portion is the mucosa. And mucosa get what? The epithelium. This portion is the lamina propria, and ending up with the muscularis mucosae, isn't it? So it's being said that the glands have got pits on it. The opening region through which the gland has to pour its secretion on the surface, right? So they have got deeper pits. Upper 2/3 of the mucosa is showing the deeper pit, and the lower secretory part is in the lower 1/3. इसका मतलब है 2/3 pit है, 1/3 region इसका secretory element or base of the gland address कर रहा है, right? Yes. And the variety of the gland is simple tubular or simple branch tubular, but the lower end is going to be coiled. So आपको इन्हीं के cut sections ये मिल रहे हैं, right? You get that how histological features comes in कि आपको ये LS में दिखाई दे रहा है, और अगर इनकी basis जहाँ कटी हुई, you observe the oblique or rounded section. Is it clear?
Yes.
Now we will see some other picture to show the pyloric and the fundic gland. Okay, this is the region of the pylorus. When we cut it, what we see again, the deeper pits are again there, and the basal region is lower 1/3 to show the secretory element, right? Again, simple, branched, tubular, but can be coiled, right? So in the cardiac and the pyloric, we get the pits which are deeper, occupying the upper 2/3, and the secretory element or the basal region is in the lower 1/3. Same thing. So let's see if there is any difference in the body of fundus. Now you can see what? That here the pits are shallower, occupying the upper 1/3, but the lower portion, the more secretory elements are occupying the lower 2/3. Is it clear? Simple to remember that the cardiac and the pylorus, they have got deeper pits, and the body and fundus region will admit the shallower pits, right? Okay. Now we will observing the structure of a gland. Concentrate here. If these are the pits, because we can see with the help of hand lens कि जैसे छोटे-छोटे सुराख हो एक surface पे, they will be showing the pits. अच्छा. अब ये portion जो नीचे की तरफ move कर रहा है, that will be addressing the portion of these are the surface cells. Then comes this region, the isthmus. Then comes the neck part, and this is the base. So we can learn that the parts of the gland are its isthmus region, the neck region, and the basal region, right? So let's find some other picture to concentrate on the detailed features. And there is a very unique thing about a stomach, that being a hollow organ. What other hollow organs lie in our body? Can you name any? In addition to your stomach dilated? Intestines, tubular portion में आ जाएगा तो more the bladder, ठीक है? दोनों तरह के bladder, the urinary bladder, the gallbladder, and the uterus. So they all hollow organs are having the random arrangement of the muscle, right? But the most random arrangement of the muscle is in the stomach region. So let's see that if we can observe a particular layers here. So you told me first the inner one was the mucosal layer, then comes the submucosa, then muscularis externa. So focus on the muscularis externa, which is being shown. The innermost layer of the muscularis externa, definitely they all are made up of the smooth muscle variety. It is lying in the direction of inner oblique, obliquely set fibers are there. Then you see the middle layer, which is circular. The muscles are arranged in a circular fashion, right? And then comes the longitudinal layer. Which one now has become the outer layer? So, do remember inner oblique, middle circular, and the outer longitudinal. These longitudinal layer, they are well developed against this lesser and greater curvature, but along the anterior and posterior aspect, they are not well marked, right? So do remember, सुन्नत है जी कि हमने बैठ के खाना पीना है. So what is being taught? That if we eat up or drink any fluid, any liquid, while lying down, our posterior wall of the stomach will get hit. So being not well much on its outer side covered by the muscular layer strongly. So it's better to sit up and drink so that the all content of the food should be present along this the greater curvature. Another safety feature you would say. Okay. Now after that muscularis externa, we'll find some other detailed structure which we can see. Okay. If we see from the above, the surface view of that stomach, so many rugis which are foldings, and a particular portion, if you focus, you can see the elevated areas. These are the gastric or mammillated area, and they are being separated by various gastric grooves, right? So when you study what mammillated area are, you should be seeing that they appear like this. Okay, some other diagram to see it in a longitudinal flow, that how they appear. So on the top you see these elevated mammillated areas and the grooves, and along with these grooves you see these gastric pits, right? And pit are the area where the glands are going to be opened. You see this tubular part, and that is being divided. So it means we are observing the muscularis mucosae at the lower end, and this is whole is the mucosa. So the self-explanatory term that if somebody asks you what is gastric area or mammillated area, what it is, and the gastric pits are also called as foveolae, right? Okay, now we have to concentrate on the cellular detail. Let me find some good picture. Easy as well. The name you took regardingly. Okay, layers को देख लेते हैं. चले इस schematic representation में again the mucosa showing the mammillated area, and you can see these are the pits or foveolae, right? And you can see the passage of the glands being bifurcated, simple branch tubular. And where lie the lamina propria? Lamina propria is here, intervening in between these bulk of glands, right? Well laden with the neurovascular bundle, right? In addition to lamina propria kaha jaye, serosa kaha jaye, submucosa kaha jaye, always the connective tissue rests in, either loosely or somewhere you get them densely. So all the features of the connective tissue that you should name some cells like fibroblast, smooth muscle cells, plasma cells, lymphocytes, so on, right? Then you can mention the connective tissue fibers, the collagen, depending on the region, the elastic or reticular if there is, and then the ground substance. So when the word connective tissue comes in, we will not repeat the details, but we should know what it carries, right? Yes. Then you observe the muscularis mucosae, which is looking as an interwoven layer, the inner circular and outer longitudinal. Then you get into the submucosa, again well vascularity is being shown here, but the special thing here are the submucosal nerve plexi, which is masseter nerve plexus, is a part of your entire nervous system, right? Then you get into this musculus externa. We just saw in the whole section the inner oblique you have to draw like this, then the middle circular and the outer longitudinal. Okay, another thing that in between almost always the circular layer and longitudinal layer is showing these, you can see the yellow dot like structure. Again a nerve plexus called as myenteric or Auerbach, another part of your enteric nervous system. So the location should be clear in mind that one plexus is always in between the muscular region and the other plexus is in the submucosal region, because it has to supply your blood vessels and definitely will give branches to address the glands, right? And that plexus will address the contractility among these muscular area. So we should know the location and the function of that respected organ. Definitely the ending up structure here is the serosa. S for serosa, S for stomach. Okay, what is the difference between the adventitia and serosa? Can anybody help? Yeah, epithelium. Along with which one? Yeah, that is mesothelium. It is along with the serosa. If this mesothelium layer is not there, we call it as adventitia. Simple term because that can be asked in the Viva question. So these are the layers. Let's see another important thing related to it. It's very slow uploading, but we have to wait. Okay, that was the picture I was looking for. Okay, now you can see the typical fungal gland or gastric gland. This is the surface area having the surface mucus cell, right? And then you see the dipping down of it. Surface mucus cell is a part of your simple columnar epithelium, which is the typical epithelium of your GI tract, right? So these cells are taller in nature, and what they carry the mucus, which is alkaline in nature and insoluble, right? So well marked with the bicarb in it. Then we move in, and this is the isthmus region and neck region, and this is the basal part, right? So in this tubular portion or excretory portion, we expect the mucus neck cell being made in this light purple color, you would say, or pink color. It could be in the shape of pyramidal or triangular in shape, right? And the basal located nucleus is there. What they carry? They carry mucus again, but it is acidic in nature, or you would say less alkaline, but soluble, right? And now, what else you observe? They are basically located in the neck or isthmus region. Then you observe another variety of cell called as stem cell. These stem cells, as self-explanatory, are the undifferentiated regenerative stem cells, right? So the surface mucus cells, the mucus neck cell and the stem cell, they have got a similar lifespan. Some occupy the 3 to 5 days for their renewal, like the surface mucus cell. Then the mucus neck cell, they take 6 days, and so as the stem cell. So what is the function of this stem cell if they are present here? These are supposed to produce all these factory of the cell. So they will move upwards to regenerate all this lot, and they will also move downwards to produce all this gain, right? So we should know that what is the function of these cells. Okay, then we observe another variety of cell, the parietal cell, which are main cells present in your stomach. What they are supposed to do? They are supposed to produce your HCl, right? And they are well laden with the mitochondria that gives energy, immense energy, for the excessive production of this HCl, right? And another important thing we have discussed in its clinical app, that the intrinsic factor is gonna be produced through its rough endoplasmic reticulum, supposed to be for the absorption of vitamin B12, right? And how it looks like, could be triangular, could be rounded. A bulging cell is having the centrally located nucleus, sometimes bulging towards the lumen or occupying the area and pushing its basal region to be, this is the area of this lamina propria, right? The area around it. Next to it, we have got another important cell called as chief cells, which basically occupy the basal region, right? And they secrete pepsinogen and gastric lipase. And why they are intensely basophilic? Because they are well laden with the rough endoplasmic reticulum, and then supranuclear Golgi apparatus is there, and ribosomes are there. So giving the intensely basophilic state. और parietal cell क्यों eosinophilic है? Because of the well marked mitochondria. So you already may have learned in the first year The different organelles, they take different states,
Yes,
right?
ma'am.
Okay. Then last but not the least, there is another variety being shown in the green are the enteroendocrine cells, right? These enteroendocrine cells are again smaller cell resting on the basement membrane and could be triangular in shape, you would say, a little bit apical nucleus, and they are supposed to be of different variety. If they are absorbed, 17 type of cells are being shown, but the most important are of this enteroendocrine, the EC cell. These EC cells, which are being written on the blackboard, are supposed to produce the serotonin, right? Another hormonal thing we observe. Then there is a lot of cells called as G cells, which are supposed to secrete the gastrin. Then comes the D cell to produce the somatostatin. Then A cells to produce the enteroglucagon. In addition to it, motilin could be there, right? So many hormonal and other substances are supposed to produce through this enteroendocrine cell because it is a part of this diffuse neuroendocrine system, DNES being abbreviated. Okay, if somebody asks you कि जी आपका endocrine system कहाँ है? एक तो आप endocrine system main glands बताओगे ना, thyroid, parathyroid, adrenal. But keep in mind that stomach is one of the place where you get this collection of cells called as enteroendocrine. So these cells, where they rest, definitely they are in the lamina propria of the mucosa, but the running lower place is being occupied by the capillaries, because you always know the endocrine cells, they are ductless in a sense. If you imagine that cell, so the secretion they are going to produce is going to enter into the blood, right? So from all the stomach, ये question भी आ सकता है कि explain the cells of the stomach, right? या stomach gland, उसमें आप क्या लिखेंगे? Stomach का आया तो आप ये 6 सारे सेल लिख दोगे surface mucus के साथ, और अगर gland के सेल आए तो you will leave the surface mucus cell, you will start with the number 1 mucus neck cell, stem cell, then parietal cell, then chief cell, then enteroendocrine. अच्छा, इनका लाइफ स्पैन हम डिस्कस कर रहे थे कि स्टेम सेल का, म्यूकस नेक सेल का और सरफेस म्यूकस का तकरीबन अ वीक है. कोई 3 से 5 दिन लेता है, कोई 6 दिन लेता है रफली, राइट? Then comes the lifespan of parietal cell. The most important है, they take almost 60 to 200, 150 to 200 days, I think it's being written, right? And then the lifespan of chief cells and enteroendocrine 60 to 90 days. So easy to remember कि 3 सेल के आपस में मिलता जुलता लाइफ स्पैन है. इन 2 सेल्स का सिमिलर लाइफ स्पैन है, सिर्फ parietal सेल का सबसे ज्यादा लाइफ स्पैन है. So now we will see some of the individual cell detail, because again it's important, like parietal cell or so. These are the histological view. Let's see first then. Okay, if we place the pyloric stomach under the microscope, you can see the glands running down and showing the branch pattern, and the intervening area is the lamina propria. This region is the muscularis mucosae, so this is mucosa. And these folding, these are called as rugis, right? And this area is the submucosa, and keep in mind that there are no glands in the submucosa. All the glands are in the mucosa lining. Then we enter into the muscularis externa. Okay, now let me show you the details of the individual cells, because सब छोड़ के किसी एक सेल पे नोट आ जाएगा, then again it will produce a little bit difficulty, so we have to see them one by one. We see the distance from the incisor teeth. ताकि हमें रफ आइडिया हो कि आपकी ट्यूब स्टमक तक पहुंच गई है कि नहीं, आप कहीं और गलती से किंग तो नहीं कर गए. So the first place is the pharyngoesophageal junction, which is 6 inches, you would say, from the incisor teeth. Then when the tube moves up to the 9 inches, that could be the area of the arch of aorta. When it passes up to 11 inches, there could be the principal bronchus junction. The trachea is going to be bifurcated into the two principal bronchus, right? Then when you are supposed to see that 15 inches has been passed, so that could be the area where you have passed through the diaphragm, right? Crux of diaphragm. So that was about the inches distribution along with the esophagus. One of the clinical application, you would say that it's important for learning sort of purpose. So after that we have to definitely learn the histological features. So let's see how they are looking. Being a tubular structure, when you cut a tube transversely, और वो आपको transverse view दिखाया जाए, तो how it will be appearing? The mucosa is thrown into these foldings. When empty, ये आपको इस तरह collapsed, crumpled दिखाई देगा. जब आप खाएं पिएंगे, the things are supposed to pass through it, the food, it will be dilated. So this is the mucosa. First comes first, and then comes the submucosa, right? And this portion, the lighter area is the submucosa. Then this is the muscularis externa. Then we enter into the adventitia. And before the esophagus has entered into your diaphragm, it is being lined with the adventitia, and when it gets entered into the abdomen, I think most probably it is being lined with the serosa, right? So that is the histological view you observe in the esophagus, the 4 layers. अब आपको ये 4 layers याद रखनी है पूरे GIT में, कि आपने इन 4 layers की heading देनी होती है, फिर एक लेयर को पकड़ लेना होता है कि म्यूकोजा, जिसमें एपिथेलियम, लेमिना प्रोप्रिया और मस्कुलरिस म्यूकोजी. फिर सब म्यूकोजा पे आ जाइए, उसके सारे फीचर्स बताने होते हैं. फिर मस्कुलरिस एक्सटर्ना कैसा होता है, और फिर एंडिंग अप विद द एडवेंटिशिया सेरोसा. सो आपके मार्क्स कहीं नहीं गए कि एटलीस्ट आपने हेडिंग डाल दी है, और इंडिविजुअल आप डिस्कस करने लगते हो तो देखते हो ना कि जिस तरह चीजें तस्वीरें अपलोड होती हैं, दे टेक टाइम. तो जो चीज खोलते रहोगे वही खुलेगी. तो जब आप लेयर्स के नाम डालोगे, म्यूकोजा को कंसंट्रेट करोगे, आपके ज़हन में आएगा एपिथेलियम लिखती थी, फिर आपको वैरायटी क्लिक करेगी, फिर आपके ज़हन में आएगा कि कुछ ग्लैंड्स भी बन रहे थे. जब ग्लैंड्स में पहुंचोगे, यू विल रिकॉल कि कुछ सेल्स भी थे. जो सेल्स के नाम लिख लोगे, लाइफ स्पैन, उनका फंक्शन, उनकी प्रोडक्शन, उनका न्यूक्लियर, उनके ऑर्गेनेल. सो दिस इज द वे दैट हाउ यू रीच टुवर्ड द सब्जेक्ट. अदरवाइज मेडिकल एक जस्ट लाइक अ सी, बड़ा मुश्किल लगता है कि यार इसमें gross takes much time. So how to do this histology and embryology? So you have the smart study work, you can do it. Now after that cross section view, we will be moving towards another section, a picture to see the detailed structures of the. Okay, let's see. So first is the mucosa. Mucosa, what epithelium I expect in the esophagus? Stratified squamous, but non-keratinized. So this is again a rule that the things that are into your internal body lining area, wherever you get the stratified squamous, it is the non-keratinized, right? So epithelium here is stratified squamous. You make the top cell layer squamous and put the nuclei in it. The basal region will be addressing the cuboidal epithelium, right? After that, you are still coming towards the next layer, which is this submucosa. But here they have made the muscularis mucosae. Okay, the muscularis mucosae of the esophagus is the thickest one throughout your GIT. That MCQ can come in this form. MCQs can be that which organ of your body among the GIT has got the thickest muscularis mucosae. Okay, so that would be the esophagus, the true option. Then when you enter into the submucosa, you see a bulk of gland, which are the esophageal glands proper, right? And if the cardiac region is being cut, you can observe the esophageal cardiac glands. So two types of glands you observe. Then the muscularis externa is there. So there is something different about the muscularis externa as well. So we'll pick some other picture for the full view. discuss the schematic diagram. That you see the mucosa lined with the epithelium, the stratified squamous, non-keratinized. Then you enter into loosely arranged thick lamina propria, and that lamina propria is showing various cut blood vessels and showing the excretory duct of the gland. They must be showing sometimes the esophageal cardiac gland if the cardiac region is being cut, right? Then you see this muscularis mucosae, the pink dots. They are being shown and they are showing the thickest muscularis mucosae. So here is the mucosa. Then we enter into the submucosa. Submucosa में आप ये bunches देख रहे हैं mucus secreting glands. They are called as esophageal glands proper. और जो आपने glands lamina propria में देखने थे वो थे esophageal cardiac glands, क्योंकि वो cardiac या esophagus स्टमक की कार्डिक एंड के करीब था और उससे मिमिक करता था, इसलिए उन ग्लैंड्स का ये वो था सपोज टू प्रोड्यूस द म्यूकस. देन डेफिनेटली यू कैन सी द एडिपोज टिश्यू, द कनेक्टिव टिश्यू, द ब्लड वेसल्स. और यहाँ पे हम क्या एक्सपेक्ट कर सकते हैं नाव प्लेक्सिस में? द मेजेनर नाव प्लेक्सिस. ये आपके ज़हन में होना चाहिए कि सब म्यूकोसा आपने लिखना है तो उसमें एंटेरिक सिस्टम का मीजनर नाव प्लेक्सिस मेंशन करना है, वो आप लेड पेंसिल से ड्रॉ कर सकते हैं टू रिमेंबर. Okay, then comes the muscularis externa in the form of inner circular and outer longitudinal. In between, you can make the myenteric plexus or our breast plexus, right? Okay, then comes the adventitia. You put the neurovascular bundle, the adipose tissue, the connective tissue. Okay, the important thing here about this muscularis externa. For this, I have to move towards the gross picture again. Something important in relation to the muscularis externa of the esophagus. That is hopefully the last thing of it. Okay, we can take if this section. So we will divide the esophagus. Kindly concentrate. This is again important. We can divide it into upper 1/3, middle 1/3 and lower 1/3 in 3 parts, right? So this upper 1/3 muscularis externa will be showing the skeletal muscle. Simple. Why? Because the skeletal muscle was in the this pharynx. तो जो चीज आपके pharynx में थी अल्लाह ताला ने वही कंटिन्यू कर दी upper 1/3 of the esophagus. Middle में क्या हुआ? There is a mixing. The skeletal muscle प्लस स्मूथ मसल्स, राइट, मिक्स हो गए. लोअर 1/3 पे आ जाइए, इसका मस्कुलर एक्सटर्ना काटोगे तो यू विल ऑब्जर्व द स्मूथ मसल ओनली. क्यों? वो स्मूथ मसल फिर बाकी पूरी जीआईटी में, चाहे फिर वो आपके पास स्टमक हो, चाहे स्मॉल इंटेस्टाइन या लार्ज, यू गेट द स्मूथ मसल. सो द नेचर इज अकॉर्डिंग टू द रूल्स और द कंपनी यू कीप. अपर में स्केलेटल क्यों थे? क्योंकि इनकी कंपनी फेरिंग्स के साथ थी, तो स्केलेटल मसल. लोअर के पास प्योरली स्मूथ क्यों थे? क्योंकि इनकी कंपनी आपके स्टमक के साथ ही जिसमें स्मूथ मसल है. और दरमियान में मिक्स क्यों थे? क्योंकि इधर से स्केलेटल आ रहे थे और इधर से स्मूथ. सो सेंटर विल एड्रेस द, इट डिपेंड्स कि जब आप स्केच बनाओगे और इसोफेगस व्हेन यू विल राइट और ब्रैकेट में विल राइट दैट इट इज अपर 1/3 और मिडल और लोअर. तो ख्याल रखना अगर अपर लिखोगे तो मस्कुलर एक्सटर्ना सिर्फ स्केलेटल बनाएंगे, अगर मिडल लिखेंगे तो दोनों बनाएंगे आप, और अगर लोअर लिखोगे तो सिर्फ स्मूथ. सो द थिंग स्पेशल अबाउट द इसोफेगस इज दिस. और हमने स्पेशल इसके स्टमक के मस्कुलर एक्सटर्ना की क्या सीखी थी? द 3 लेयर्स इन डिफरेंट डायरेक्शंस, लेकिन थे सारे स्मूथ, राइट? इट्स क्लियर? ओके, अब इसकी स्लाइड देख लेते टू एंड अप द टॉपिक. The schematic things are definitely much clearer, but on the screen, the slides we are supposed to. So this is the area which is called as the mucosal epithelium stratified squamous, non-keratinized. After that, we move into the lamina propria, which is much wider. Can you see that? Lamina propria, या अगर note आ जाए कि discuss the gastroesophageal junction, ये भी note आ सकता है. क्या करोगे? एक तरफ स्टमक लिख लोगे, एक तरफ आप इसोफेगस लिख लोगे, और उधर पैरामीटर्स में एपिथेलियम से स्टार्ट कर ले, मिकोजा की एपिथेलियम। इसोफेगस में एपिथेलियम क्या है? स्ट्रेटिफाइड स्टोमस नॉन केराटिनाइज्ड। उस तरफ क्या लिखेंगे? सिंपल कोलमनार। अच्छा, यहाँ लिखेंगे लेमिना प्रोप्रिया इज मच वाइडर, और उसमें क्या हो गया? स्ट्रीक्स की तरह पतला हो गया। ठीक हो गया। अच्छा, स्टमक में ग्लैंड्स बहुत मैक्सिमम अमाउंट में है। यहाँ पे आपको पर्टिकुलर रीजन में मिलेंगे कि इसका कौन सा रीजन नियर टू द कार्डिक एंड है, तो यू विल गेट। आई एम स्क्रॉलिंग एंड स्क्रॉलिंग। दिस इज द excretory duct line with the stratified stomach non-keratinized epithelium towards the surface. Rest of it is not addressing here any other thing. So it means that couldn't be a part cardiac region नहीं होगा. Otherwise you get the esophageal cardiac gland. Okay. इसकी हमने ये जो आपको ये bunches दिख रहे हैं बहुत सारे, ये अगर आप मस्कुलर मिकोजी को एड्रेस करें तो thickest one हो जाएगी. So we are now supposed to enter into the सबमिकोजा में आ गए हम. अच्छा, कोई लाइन नहीं खींची होती. कीप इन माइंड कि मिकोजा खत्म हुआ तो कोई फुटे से लाइन होगी. नो, you get those portions of musculus mucosae, then you jump up into the submucosal connective tissue layer. Here what you get a collection of bunch of these mucus secreting acini. These are esophageal glands proper in the submucosa, right? Okay, then you enter here. This is the muscularis externa. If you see simple smooth muscle, that is the lower, but here you get the skeletal muscle. Can you see?
Yes, ma'am.
These are the skeletal muscles with the periphery. It means we are in somewhere upper 1/3 of your esophagus, right?
Yes.
That is why we are unable to see the esophageal cardiac glands, क्योंकि हम ऊपर वाले रीजन में थे. तो ये देखिए थिक लेयर है अरेंज. ये इसका कट सेक्शन, ट्रांसवर्स सेक्शन भी आपको बता रहा है कि देयर इज द skeletal muscle because the periphery is addressing the nuclei. These bunches are here, and finally we reach up to the ending the adventitia. There is no mesothelium here. So this is the way to identify a slide. So once you are able to learn, then it becomes easy. These are the adipose tissue intervening. That could be a portion having various ganglionic cells. Could be the mentoric sort of nerve plexus, right? जी.
इसमें स्मूथ मसल भी है.
स्मूथ मसल्स आपको किस जगह पे दिख रहे हैं? अगर एक्सटर्ना में मिल जाए तो मिडल रीजन होगा. ठीक है. अगर आपको ऊपर बेटे मिल रहे हैं तो मस्कुलरिस मिकोजी होगी, राइट? मस्कुलरिस मिकोजी everywhere in that esophagus will be showing the smooth muscle. तो अगर एक्सटर्ना में आपको मिल गया है तो डेफिनेटली मिडल रीजन स्टार्ट हो गया, चाहे उसका इनिशियल कंटेंट हो या उसके कहीं दरमियान.
तो ये जो मैम इसके बिल्कुल साथ है.
ये जो इर्द गिर्द बेटे ये जो चीजें होती हैं, ये सारी की सारी जो फाइबर्स हैं, ये आपको कनेक्टिव टिश्यू बता रही हैं. ठीक है. और 40 पे ले जाओगे तो विल बी एबल टू. तो डेंस कनेक्टिव टिश्यू की फाइबर्स भी आप देखते हो ना, वेरी डेंसर अरेंज होती हैं. और कुछ आपको ये जो राउंडेड सेक्शन दिख रहे हैं, ये ब्लड वेसल्स हैं. ठीक है. और एपिथेलियम हम यूजुअली एग्जाम में जैसे आपके लिए फोकस करें, वी आर ट्राइंग टू फोकस अ स्लाइड विद द स्ट्रेटिफाइड स्क्वेमस, और हम कुछ सर्च करेंगे कि जिसके नियर बाय. लेट मी मूव इट टुवर्ड्स द 4x एज वेल. थोड़ा सा आपको बर्ड्स आई व्यू मिल जाएगा, थोड़ा सा ज्यादा एपिथेलियम और फोल्डिंग क्लियर हो जाएगी. अब आप देख रहे हो तो आपको पता चल रहा है कि ये मस्कुलरिस मिकोजी है. यू गेट, दिस इज एपिथेलियम, दिस इज लेमिना प्रोप्रिया, और ये थिकेंड पूरा ये जो पट्टी की तरह जा रहा है, दैट इज मस्कुलरिस मिकोजी. फिर आप सब मिकोजा में आते हो. ठीक है. जब हम 10 पे 40 पे जाते हैं तो वो आपका जो एक दूर से इमेज है वो लॉस्ट हो जाता है, इंडिविजुअल स्ट्रक्चर पे फोकस करते हैं. ठीक हो गया. सो द वे टू आइडेंटिफाई हाउ वी. अब ये देखिए स्केलेटल में आगे, अब बीच वाली एंड तक अगर आप चले जाएं, अब सब मिकोजा को रोटेट करके हमें ढूंढना पड़ेगा कहाँ हमने वो बल्क देखी थी. ये इसोफेजियल glands proper. When we concentrate, the parietal cell can come. So what you see in that very parietal cell that is going to produce axial intrinsic factors, so and so. See towards the lumen side, what you see, there are so many intracellular canaliculi. small canaliculi are being formed by the plasma lemma inside. And what else you see, that there are so many tubulovesicular system which are membrane bound. So when it is not in an active state, this tubulovesicular system, that is well marked, but when it has to do some activity, these tubules or vesicles, they coalesce and form the intracellular canaliculi, because they have to secrete much HCl, right? And what else you see? You see this nucleus, nucleolus, and the Golgi apparatus is there, and so many mitochondria are there to give energy to this whole. And they are also lined with the microvilli to increase the surface area. And if I draw another cell here, they have got apical junctional complexes. ये तो आपको पता है कि एपिथेलियम में जो cells पड़े हुए हैं, they have got junctional complexes intervening. So that is important. So now let's see the slides of it. Okay, it's a little bit tighter, so I have to switch it off. Kindly concentrate. We are starting from this mucosa. This whole is mucosa, right? And from here, the surface mucous cell, which are showing various pits here and running inside to form the deeper glands. So ये इतने closer है, if you see, ये कुछ इस तरह से हैं जैसे आप 8-10 तस्बीह के दाने पकड़ लें and hold them up, right? And they are running downwards to form the simple branch tubular glands, right? Intervening जो closely streaks बच गई हैं वो lamina propria के लिए place है. Then we reach up to the muscularis mucosae, right? And then you come into this submucosa, जो कि cut section of blood vessels तो जरूर address कर रहे होंगे, but no muscle in it. So what is rugi? ये जो पूरा fold उठा हुआ है, the mucosa and the submucosa, this whole curve is the rugi, right? And when you eat up the thing or we fill it up, so these rugi are vanished, ये straighten up हो जाएंगे. जब ये empty है तो आपको ये transverse folds दिखाई दे रहे हैं. Okay, after that muscularis externa की layer है, and sometimes we see the scattered. This is the serosal last layer. So अब इसको identify after 4x हमने कैसे करना है? With the help of parietal cells resting on this layer. Okay, now 10x ये आप सिंपल कोलमनर एपिथेलियम देख रहे हैं और ये अंदर डीप सीटेड ग्लैंड बनने जा रहे हैं. ये जो राउंडेड हमें सेल्स देख रहे हैं, लिटिल बिट eosinophilic stain ज्यादा है. If you can observe them, you can see these are the parietal cells. और how closer, ये आप देख रहे हो कि ग्लैंड्स बिल्कुल ऐसे जैसे क्लोज पड़े हुए हैं और जो दरमियान में थोड़ी सी जगह है वो लैमिना प्रोप्रिया के लिए. So these are the typical look of the funding gland of the stomach, right? So this area could be focused under the microscope for identification. Now we will see the other region as well. This is the cardiac region. You can see again, this is the mucosa and the glands are simple, could be branched and running downwards up to the muscularis mucosae. And definitely these curves are the rugosities, right? These whole curves, ये जो एक और fold आ गया, ये सारे रूगी कहलाते हैं, right? अभी आप 4x पे देख रहे हो, इसको 10 पे ले जाएं तो कुछ इस किस्म की जो क्लियर हो जाएगी. So they are again lined with the mucus secreting cell. Here the other variety could be the enteroendocrine cells that are should be the in the lining epithelium.